Okay. Let me know when. Oh, okay, awesome. All right, so I'm going to be doing a talk today on current computational biology research. This is specifically work being done in the Noble Lab, which is under like the Department of Genome Sciences. They're super cool. I love my PI, Will Noble or Bill Noble. He's great. So yeah, this is like what's I guess different about this versus like something else is this like I'm literally like gonna wake up tomorrow and work on this, so it's very much in progress research, which is nice. So the ending conclusions aren't necessarily like, and then we can now solve this problem. It's more like, and then what? Like, you guys can even throw out some ideas, and maybe they'll end up like in the research. So I think that's pretty cool. So the title, this was originally prepared for my lab, so this is supposed to make sense, but it I don't think it would off the bat. So I'll start just breaking it down. Ignore the title for now. We'll come back to it, and hopefully by the end of the talk, you understand exactly what this means and why it's cool. So here is the background, which is actually probably where most of the talk is going to be. And please feel free to pause and ask questions if I use a term that's like not commonly known. So let's start off with like what's the data I'm working with? Like, well, literally what machine does this come from, right? Because it's computational biology research. There's some kind of like biological backing to all this, and that comes from this machine over here. So the machine that gives like the data that I work with is called a tandem mass spectrometer, and it is like a really, really ubiquitous protein content analysis method. So the short form of it is basically you have this protein soup, basically. Like, right, you start with a bunch of proteins, and then you will digest them with an enzyme, right? Standard is trypsin, right? And it cleaves before R after K or something like that. But the point is basically you have like this proteome of like some kind of organism, like E. coli, yeast, even humans, and then you digest it with an enzyme, which then cleaves up this protein sequence into a bunch of shorter sequences, which I will refer to as peptides. So if you're not familiar, like just like how there's like the genetic alphabet A, T, C, G, which is 4 letters, there's also the protein alphabet, which is 22 amino acids, right? So it's a little more confusing, definitely more complex, more information, if you will, for sure, in this alphabet. And this is one of the ways to actually figure out, okay, here's like a protein soup. What is the prevalence of this protein? What is the prevalence of like this peptide? Which can tell you a lot. It can help you find like biomarkers for like diagnostics. Like for example, if you have a diseased tissue and a non-diseased tissue, and you know that one protein or peptide is heavily expressed in diseased tissue and not in the other, well, how do you find out? You can't just look at them and like tell the difference. Maybe they look the same, and it's more of like an internal issue. You'd use a mass spectrometer to figure it out. Like, oh, this sample had much higher expression of this like specific peptide, and we can tell that therefore it's diseased, right? Or maybe you don't know what you're looking for, but you're looking for like, oh, what stands out? Like, hey, this one seems to have a huge abundance of this peptide, and this one doesn't. What does that tell us about something? It basically is like, if you want to look deeper into like what peptides exist and what proteins exist, you will most likely use like tandem mass spectrometry. This is one of the most ubiquitous methods. If anyone else knows any more, like feel free to like comment on that. But as far as I've seen, like it's used all over the place, like tandem mass spectrometry in different ways, but it is at the end of the day some kind of like mass spectrometry. What is the tandem part? Also push the this thing? All right, yeah. You still good? Okay, yeah. So the tandem mass spectrometry comes from the fact that you actually, and I'll get a little more technical here, this spectra, like the fragments are actually kind of ran through twice, basically. You have this like, you do some kind of scan, and you have like a precursor ion, which is basically a charged form of like some peptide, right? And then what happens is through some really crazy fancy technology, it's fragmented like again. And then essentially there's 2 types of ways to do it. Are you familiar with DDA and DIA data by any chance? No. I'll give the short form of it. Tandem mass spectrometry means, along with this, which is what the ultimate data form will look like, you also get some information about like precursor, which is like the peptide fragment that generated this spectra, precursor mass to charge ratio from like the first MS scan, and the second MS is like what makes it tandem. It's like this is definitely getting into the weeds here. It's not too important. You get charge information, you get some retention time, you get like precursor M over Z, which is mass over charge, the ratio, which is important. It's essentially more information, and this would mean something if you were like in proteomics and you're like, oh, tandem mass spectrometry. But for our cases, it's just like all you need to care about is basically it is very widely used. And this is what like a data readout would look like. You'd have a bunch of like X values, which are M over Z, which is mass over charge, right? And then you have intensity, which I'm actually not fully sure what that would necessarily represent. So it's a little bit like foggy for me, but it seems to be just like how heavy did this seem to appear to the machine. And this ultimately can be reduced to a bunch of points, X, Y coordinates, right? Which is what I work with. But where it comes from is kind of important because when I get into like the data and like the analysis of it, we'll obviously need to come back and refer to like, because we're also trying to figure something out about the peptides that came in and generated this, right? So I'll be, later on, this will become somewhat important. So here is how things are traditionally done, right? So what'll happen is you will know the organism you're trying to analyze, right? You will say, I did a run on some E. coli, right? And we know the proteome of E. coli. We can have like its entire just proteome in like one big FASTA file. It's literally just in the megabytes. It's not that crazy, which is kind of insane to me, right? And essentially what you do is you do a parallel to what you do like in the lab, like you do it in silico. So just like how before we took this protein soup of E. coli, digested it, shot it through a mass spectrometer, and got a bunch of experimental spectrum, which is like the thing I was showing before, like the ultimate readout of the machine. You take the protein sequence database from the FASTA, you digest it in silico using the same cleavage rule that would happen in nature with the enzyme, and then you get a bunch of theoretical spectra. Like technically, if everything was perfect and this machine had no noise, no interference, no anything, what would this peptide fragment to look like, right? And then you would basically just take these two and score them against one another, right? And hopefully, what should be true is that the experimental spectrum look a lot like the theoretical spectrum of the peptide that generated them, right? So you say you had PEPTID as the peptide that generated this thing, right? Then you would have PEPIT, or whatever I said, and you turn that into a theoretical spectrum. I'm not sure what the process is for that, but I think it's pretty just algorithmical. And then one of the most simple score functions is literally just like a sliding dot product of the two, because again, this reduces, even this is shown in the graph, it reduces to a bunch of points. And through that comparison, you can score basically each each experimental spectrum. So everything that comes off the machine, you basically score it against a bunch of candidates. Like, these could have generated this theoretical spectra. Now let's do this little like sliding dot product to figure out which one actually generated the spectrum. And you'll get this list. Like, here's a peptide, here's the score it's given. And then you pick the highest scoring peptide and say, I believe with a certain level of confidence that this peptide generated this spectrum, right?
So it's like a matching problem?
Exactly, it is a matching problem. It itself has problems, which I'll get into. Otherwise, I wouldn't be researching, right? But, yeah. So this is the standard approach to like saying we did a tandem mass spectrometry run. I want to figure out what the abundance of this protein was, or I want to figure out what proteins exist in this this experiment. That's the standard approach. Yeah, and this is just what I was talking about. One thing to note is that like you will digest the protein sequence database. You'll digest it, and you'll get a bunch of peptides. Now, it would be a huge problem if every single one of those was a candidate, right? because you can imagine like the proteome is just huge, and you split it up and you get like hundreds of thousands of candidates for each spectra, and then you have 100,000 times what another like million spectra that come off a machine, and you have to do a ton of scoring, right? Well, we kind of remove that problem to an extent by only considering peptides that it makes sense that they could have generated this spectrum. For example, if there is a spectrum that was super simple, we would expect the peptide to be relatively short and have lower MCs. So basically what this means is that you will have a list of like theoretical candidates with that are determined with a plus or minus some kind of like mass over charge ratio window. So like super long peptides that are like yay long will have like a really high mass over charge ratio, and therefore only be considered candidates for spectra that also seem to have a super high mass over charge ratio. This is where the precursor information comes in, right? Because it's tandem mass spectrometry, we have precursor information. This spectra is like 500 m over z units, and these peptides theoretically are around that range, like maybe 450 to like 550. So we're only going to consider those. The ones that are like 900 m over z units, there's no way that would have generated the spectra, so we're just going to throw it out for now, right? So it just makes the problem like possible, because the cross product of the two sets would just be like a cross product. What do you call it? Cartesian product of the two sets would be huge. Yeah.
So the thing I don't understand about this is like using the MC metric. Now you have two things that can change. So like my understanding is peptides can be all different charges. Maybe there's like a reasonable range, but you know, you could have two proteins or peptides with completely different masses and completely different charges, but they have the same MOC ratio.
So we only use for figuring out which candidates pair with which spectra, we only use the M over Z. And also, I believe the ionization of the peptides is controlled by the mass spectrometer itself. It's not like they naturally come out ionized. So it's like, I believe there's some other fancy technology there that would eliminate that problem, because that would obviously be a big problem. But I mean, the candidates don't all have to be perfect. We'll pick the right one using the score function, right? So even if there is like a peptide that has the proper M over Z, but it's just way out of whack, this will get that out of there. Yeah. So is the idea of database search like the traditional formulation to like figuring out which peptide generated which spectra? Does that make sense so far? In case it was a bit much, which it might have been, I'll just reduce it to this sentence. We have a bunch of readouts from a machine. We're trying to figure out which peptides, which sequence of letters made that spectra, right? Which is useful for figuring out which proteins were in the soup you shot through the machine, right? So that's the point. Hand designed score functions, which is like the thing I was explaining with like the sliding dot product, have issues. One of the main ones is that they're really poorly calibrated with respect to charge and also peptide length. What that means formally is that for something to be well, can you guys see that? Well, that means that for basically, let me see if I remember the exact formulation for this. For different like MZ ratios, so basically for all I and J, like basically all of like the spectra, a score of like a 5 on I should mean the same as a 5 on J, no matter what, right? Even if let me rewrite that. If I has like charge +3 and J has charge +1. This is what we want. We want that no matter what the charge is and what the peptide length is, a score of 5 means a score of 5. But that's not the case. You can kind of see that here. If you have the peptide length, here are different graphs for the peptide length, and all you need to really worry about is this like little blob of red over here, you can kind of see that for lower peptide lengths, the distribution of scores for like the wrong ones is just it's just different. And you can also see that for the target ones, like a score here for peptides from length 0 to 10 is like, it's okay. It's like where most of them lie. But when you get down to peptide length 30, that same score is abysmal, right? Because the targets, which are the proper ones, are like over here. So like, if you have a score of like 10, it doesn't tell you immediately if that spectrum is good or bad. You also have to consider some other like orthogonal factors, which is less than ideal, right? So that's one problem that hand design score functions have. Another problem that hand design score functions have is just like they might not have the most like statistical power. We would like to see a really, really clear separation between the blue and the red groups. Basically saying when something is right, we know it's right. We're very, very confident it's right, and the score is like way higher. And then when something is wrong, we know it's wrong and the score is shot way down. But that's not really the case. You can see that there's stuff in the middle, heavy overlap between some of the blue and some of the red. Basically meaning that we're not sure a lot of the time. Like something's kind of in between. And that doesn't bode well if you're trying to be very sure about something. It's like it's got a score of 10. That seems high, but we don't know if it really is because of the calibration issue. And we're not sure if it's high enough to say that we're confident that this is an actual pair, right? So that is one of the main like problems with like these hand design score functions which I have listed up there. Some of them, they're just names. Don't worry about them too much. And there's this little paragraph at the bottom, and this is probably one of the more complicated parts of the talk that I'm giving right now. It took me a while to understand like how statistical control is performed for like tandem mass spectrometry experiments. It's really critical because otherwise you'd be saying, oh, there's a bunch of this peptide in this soup, and it's not there. Like it's straight up not there. Like the database search thought it was there, but it just wasn't. There has to be some kind of way for us to verify that the matching was done properly, because there is no ground truth here. We don't know the ground truth. There's nothing to really compare it to, so how do we do that? That's a problem that there is a solution for, at least to some extent. So there's this method called target decoy competition that is very heavily used in this space, and it's basically like what can let you get peptides and spectrum matches. PSMs is what they're called, with very high confidence. Does everyone know what I mean when I say PSM? A readout from the tandem mass spectrometer paired with a sequence of letters, and you being like, that's correct. Like this peptide sequence generated the spectrum. That's a PSM, peptide spectrum match. Okay. How is the understanding so far? I've really not come to any great conclusions yet, so this is all just background. So if you're like, that makes sense, and then we should be okay. And please stop and ask for any questions. Anything? Okay, cool. So I'm going to go over this in a very high level because it's not super important. But and I think this is just a cool thing to think about in general because it's not unique to proteomics and could be applied in other places. The concept of target decoy competition. Basically, it's like we have a bunch of peptides, like 20 maybe, or to 100 per spectrum, right? And we're trying to figure out which of these peptides actually made the spectrum and which of these guys are like phonies or like actually didn't make them, right? So we don't know which ones are right and which ones are wrong, right? But what we can do is make ones that we know for a fact are wrong, right? And call those decoys. So we have like some sequence like WALI, something like that, and then a P, and then this is like, okay, this is in the proteome. It's theoretically possible to create because there was like a K before it and then like an R after it. So trypsin would snip at these sites and create this peptide when you're doing the digestion. But we know for a fact that PAILW or something like that would never exist. It doesn't exist in the proteome, right? So now we have something that is verifiably true and verifiably false. And what we do is we put these false ones into the search as well, right? And now we know, basically, and here is where some assumptions come in that I'm not going to go over because they're very statistical. But the general idea is that the rate at which you match to decoys, the wrong ones, estimates the rate at which your discoveries are false, right? So if we match to, if 1 in every 100 of these matches, you say like you have one decoy and you say this is the right one when we know it's not. Like the score function thinks it's right and puts it at the top of the list, but we know it's not correct because we are able to mark this as a decoy because we made it ourselves, right? When we match to one of those, then we know that, okay, considering that that one was wrong, another one of them must have been wrong. We're probably erroring at the same rate. And this has to do with the fact that the distributions of these decoys and distributions of the targets are the same, and also how the score functions work. But that's not something I want to go into like super clear detail about. But the general idea is we don't know what's true and what's not. Let's make something that is verifiably false and verifiably true, and use how often we err on the stuff that's verifiably false to estimate how often that we're erring on the actual data, right? Because at the end of the day, we're never going to report this as a proper match. It's once you're done with the experiment, you throw out all the decoys and everything associated with them, right? But now you know, like, I had 1,000 peptides in my database. I matched to like 10 decoys, right? You throw out all the decoys at the end of it, right? So those matches don't really affect your final results. But what happens is that now you know that, like, okay, maybe 1% of the discoveries that I thought were true are actually false, right? That's what target decoy competition lets you do. And I'm sure it's applicable to other things with like the same like statistical distribution. So it's a pretty neat thing. I really haven't heard of it in any other space except proteomics, but I'm sure it could be used somewhere else like this. So pretty neat idea. Yeah.
And this is to test the scoring function to see like, hey, how accurate or how bad is our scoring function?
Exactly. It's to test. Yeah. It can be used to test the scoring function. It's also just, well, it's not like if the scoring function fails, you throw it out because those are still used. It's more like, yeah, like if you match to like a bunch of decoys, then you're like, well, these spectrum are garbage, right? They tell us nothing about the actual nature of the proteins that were digested and stuff like that. So what you ultimately get at the end of the day is a number of peptides and a level of confidence, which is like any good statistical test. I've identified 1,000 peptides at 1% FDR, meaning that I know that about one, like 10 in every like 1,000 of these is wrong, right? Which is like pretty good. That means that you're mostly right. So just it's a statistical confidence measure, and this is the formulation for it, right? So, let me go through this. Yeah. I won't get into the math too much, just the general idea of targets versus decoys. Hopefully, does that make sense to everyone? The idea, yeah, okay, cool. And if you like this, you could digest it later. And the last thing I'll go over is this tiny little thing over here that'll come in handy later when I show all the plots, because you'll see this thing called like Q value on the x-axis. It's a little convoluted, but essentially the FDR, it's an equation with respect to some kind of score threshold. So it's like if I set my score threshold to 10, reject everything below it, accept everything above it, what's my FDR? Right, so it's like above this score function, there's 1,000 peptides, and only 1% of them are going to be wrong, right? That's what we want to see. And the Q value is a little different than that. Basically, it's like the minimum FDR threshold at which a peptide is accepted. So you'll have a list of like peptides. I'll just do like A, B, C, D, blah, 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 right? And let's say they're ranked in terms of score. So high scores up here and like low scores down here. They'll each have some kind of Q value associated with it. And the very first one will have like 0.0001. Basically, you gotta be so strict to reject this peptide, it's not even funny, right? And then somewhere along the line, you'll get 0.01, meaning that everything above this 0.01 has like a 99% chance of being true, which is ultimately what we want to plot, right? Basically, it's like at each of these Q values, how many peptides do we accept? So then you can clearly see, it's like, I want to be really sure about this experiment. All right, go on the x-axis until you hit 1%, and then go up on the y-axis, see the number of accepted values, and that's how many peptides you have. And say you want to be a little looser, I can go up to 10% incorrect peptides. You can keep sliding on the x-axis, and then you'll see, oh, I have even more peptides, but less confidence about each one. That's what you'll get. So the idea, and this is where I get into like the actual research I do, can we learn a score function? Because so far all these previous score functions were purely algorithmical. You're given a spectra, you're given a peptide, it's some like calculation, multiplication, something very simple to calculate the score from them. But can we learn one using some advanced like deep learning techniques? And how well would it perform on some data? How can we check that, right? Would it have better separation between the targets and decoys? Would it score targets really high and decoys really low, therefore making it super easy to pick an FDR threshold? And the gold standard generally is like 1% FDR. So at 1% FDR, we would get 10,000 peptides accepted instead of 1,000 because we're able to differentiate between them so well, right? So the decoys never bleed over. That's the hope. Any questions on the background so far? You can even be like, I didn't get that. Make sense? Okay, cool. Please just raise your hand if you have any questions. Also, if you're over here, make a noise or something, because I can't see. De novo sequencing. So remember at the very beginning, I was like harnessing a de novo sequencer for mass spectrometry database search. I'm going to go over two of those words now, right? So this space is so huge, it's not even funny. I feel like I'm swimming in an ocean, but it's like it's super cool to hear like all the stuff you can do with it. Remember I mentioned database search is like the accepted way to do like figure out what was in your mass spectrometry run. Well, sometimes you can't do that, right? Because it requires that database. You need a database to perform database search. Crazy, right? So it says right there, the drawbacks of database search is you need to know a priori which peptides might occur in the sample, which is easy if you're just doing a run on like mouse data, yeast data, tomato data, like yeast. I said yeast already, but like something like that. You know the proteome of the organism which you're analyzing. But in some cases, like immunopeptidomics, you don't know all these little things that exist on the outside. You want to analyze those proteins, which would be very useful, but you can't because you don't know them because they're ever changing and such. So that's a difficult task for this. You don't have a database for that. There's also metaproteomics, which sounds really cool, but it really means you like scoop up a bunch of like shit from the environment. Like you take a sample of seawater, like a bunch of dirt, and you run analysis on that. So you got a bunch of organisms there, and doing a database search on that would result in like hundreds of thousands of candidates per spectrum, and it would take way too long to be impossible, right? That's another reason where database search doesn't work. But you can clearly see how these are very useful, right? You want to analyze a sample of the environment to see what's going on there, or you want to do something regarding like immunopeptidomics. It's like, how do we do this? What's the solution? Then this is where de novo peptide sequencing comes in. This basically says that instead of like using a theoretical spectrum and an experimental spectrum, we just take the experimental spectrum and try and predict the peptide that generated it, just from the data itself. You're not doing any kind of comparison. You're not having any kind of candidates. You don't need candidates. You just take the spectra and guess the peptide, right? Which almost seems simpler, but this is more of a recent advancement. Originally, it was done with dynamic programming, which, can someone define that for me, please? Like anybody. Yeah, fancy stuff.
Caching your solutions to problems so that you can look back and say, hey, I already did this. I already know what the solution is, so you can use it again.
Great, what he said. So yeah, little jab at dynamic programming. It's basically there were some like complex techniques to do this, and it really wasn't until the advent of like deep learning it came through that this was like more practical or it would give you like enough high power to analyze the metaproteomics and like immunopeptidomics data in this way. So then it becomes maybe a little more familiar of a problem, sequence to sequence translation. You have a sequence, a spectra, and you're trying to predict a sequence, a bunch of peptides, two different languages. It's a translation problem, which it works in the deep learning context. I'm sure Carter could elaborate with NLP and stuff like that, like exactly what sequence translation entails. But I'm just introducing this as like a proteomics problem. So like now spectra to peptide directly. Does that make sense? That's what de novo sequencing is. Right. So why does this matter? The lab has this tool called Casanovo, which is currently the state-of-the-art de novo sequencing tool. It's super cool. The PhD student that made it, Mela, he's awesome. He helps with a lot of things. So I won't get into the details of how exactly Casanova works, but it's a big transformer. It takes in the spectra, and then it will also take in some additional information about the precursor, like the mass to charge ratio and such, and then it will do like, what do you call it, autoregressive decoding on the output, like peptide. So here's an example of it. Actually, I'll go to the next slide for this one. So for example, it's like here is a spectrum, MS2, same as MS/MS, just another way of saying it. Some precursor information, which is gathered through the run. So we have all this. We've always had this. And then we also say, what are your previous predictions? And then please give us like the next token, right? So essentially, now we have something that can take in a spectra and then give us a peptide from it. And it's through deep learning. It's wonderful, right? But I'm not working with de novo prediction. I'm working with database search. So why is this relevant? It's almost like just a different problem domain. Well, the reason is we can take a look at how Casanovo, which is this tool, predicts token by token and kind of hijack it to kind of get an understanding of how it thinks peptides should be generated from a spectrum, right? So essentially, this is how Casanovo normally works. And then I'll get into the changes that I made to it and what that does for us. So originally, it just sees a spectrum. It doesn't know what generated it, right? Let's say we do have like one label pair from like a really, really, really minute, high-powered experiment. So we know this is true, but Casanova doesn't know it's true, right? So we give it the peptide and say, hey, can you please tell us, we give it the spectrum and say, can you please tell us which peptide made the spectrum, right? And it'll say, given the information and some start token, what do you think is the next most likely token, right? And it'll say, oh, I've seen something like this before. I think the first amino acid is W, right? And then I'm going to say, okay, given that the same spectrum information and also the fact that you just predicted a W, what do you think comes next? And then, if all goes well and it's able to do its job properly, which currently it is the best at doing its job, it'll give us the peptide that generated the spectra, which is de novo sequencing, right? But here's where we can do a little bit of like, I guess, hacking. It's not terribly complicated. But the idea is now we have a database search problem, a spectrum, and a bunch of candidate peptides, right? So this is going back to like one of the first slides. This is the database search problem. A spectrum and a bunch of theoretical candidates. We don't know which one is the right one, but we're going to score them all and figure out which one comes out on top, right? So this is where we do something called teacher forcing, right? Basically what we do is we say, hey, Casanovo, here's a start token. Not what do you think is the next token. We're going to ask it, what do you think is the probability of this token? We know the peptide that we're trying to score. We're not asking Casanovo what it thinks the next amino acid is. We're asking, what do you think about this amino acid I'm giving you, right? It's the difference between like me asking you to like say the alphabet. Like, okay, say the alphabet like A, B, C, D, F, G. It's like, do you think A comes next? Do you think B comes next, right? And, you know, if you've passed, kindergarten? When did we learn that? Way back when. So if I ask someone and it's like, okay, please tell me, it's the difference, like, please tell me the first letter of the alphabet, and they say A, right? That's different from saying, is C the third letter of the alphabet? You see how there's like a little bit of a difference there? I'm giving you the option, and I'm asking you, what do you think about it, right? And that's what we're doing with Casanova, right? Through training, it's already learned implicitly, like in its model, like pairs of spectrums and peptides. Like, oh, when I see this kind of pattern, I generate this kind of peptide. And now we're trying to like elucidate that information from it and use that to score spectrums and peptides, right? And here is where it can become useful, right? Say we have this green thing is a target, right? So this actually is in the proteome, it's in the database. This red one is a decoy. We shuffled it, and it's not in either of those things. This one should get a really high score, and this one should get a really low score, right? Because this is not like biologically plausible or yada yada something, right? And this one is. So notice how the first two letters are the same. So Casanova will be like, yeah, I think a W goes there. I'm going to give that a score of 0.99. Yeah, I think, and an N comes next. I'm going to give the score of 0.95. But then it's going to see a G, and it's be like, I would not put a G there. I would put a Q there, right? Because that's the correct peptide. So I'm going to give this a super low score. And now we have some way to tell that this is the wrong peptide. Does that make sense? Yeah. So that's the cool part. Now we're kind of hijacking its internal understanding of spectrum and peptide pairs for our own benefit. Yeah, that's what I just said over there. Ultimately, what happens is we will get for each peptide a bunch of amino acid scores. How plausible is that amino acid at that position, given this precursor information on the spectra? And then as you can see, obviously these ones have like low values in them. And then through some kind of aggregation, we can get ultimate scores for each peptide, targets and decoys. And as you can see for this one, it gets low, this one is high. That is a database search score function. So we repurposed this de novo. It's really ironic and cyclical if you think about it. It's like this tool was developed for a separate application, but then we kind of brought it back to its roots to see if it's learned something. Right? It's super interesting. And making this was very fun. Taking the deep learning class helped a lot because I had to vectorize one of the functions. That was like slowing it down a lot. And I didn't realize, holy shit, vectorization helps so much. It's not even funny. It literally made it possible to write a paper about this. So that's really cool. Always vectorize your code. So basically scoring this thing before I just had it like do it like, oh, yeah, there's just a bunch of like whatever I could do to remove loops in one of like the scoring things. Yeah, exactly. And that took a runtime of 1,000 hours like down to 3. So it's pretty crazy. Yeah, always vectorize when you can. If something's slow, it's probably because it's not vectorized. Yeah, and now that I've kind of gone over the implementation of the background, does it actually work? Because this is all theory so far. That's my job. So I'm going to go over. This isn't too important, honestly, so I'll get past it. But like, we chose to go over E. coli, human, and yeast data. This means something to some people. It's the machine used in the type of tandem mass spectrometry data acquisition method. Data dependent acquisition. It's I would call it standard. And then we made sure that there was no train test leakage because these are all new runs post the cutoff for the data that the original model was trained on. And we also chose to compare against 3 other score functions that are very commonly used. XCore, which stands for cross-correlation, Hyperscore, which is a fancy version of cross-correlation with some other information in there, and this thing, which I hate because I don't want to deal with that search engine. It's ugly. So yeah, this again it's not really necessary. This dude helped me with a lot of things, so thank you, Bo. Shout him out. Yeah, if it wasn't for him, I would not have gotten this. This is all like hieroglyphics to me, honestly. Like he helped me. Like proteomics is a whole other just there's so much stuff to learn about it. So he helped me a lot with making sure. Basically, what these 2 slides are making sure the experiment is very replicable, so that like if someone else wanted to verify, they could verify with the exact same database search setup and like parameters that I did. Right. It's also to make sure that across the different search engines, that what I'm actually searching for is the same. So we don't say, this search engine failed. Well, it's because you gave it a bad search, right? Maybe it could have done really well if you had changed the parameters a little bit. So we set them all to the same. All right, so this is the actual graphs. This blue line right here is, you can see the Q value right here. This is where you kind of care about it. And you can see that we actually do better than the other database search engines at 0.01 FDR, right? So this is where stuff starts mattering. What this means is that if you were to say, give me all the peptides, right, that you found that only have a 1% chance of being wrong, we would give you 6,000, and then they would give you like around like 5,000, right? So we give you more peptides. That's like a huge bonus, right? Well, yeah, it would be. Does this graph make sense so far? Yeah. So we want our line. Basically, I mean, it's kind of intuitive. If line higher, better, basically, right? So we want this Casanova line to be above the rest, right? But here's where it gets a little bit tricky, right? It's not significantly higher, and the runtime is an order of magnitude or so higher than the rest. So how else do you justify, and this is kind of like part of like the experimentation paper writing process, how do you justify and like show to reviewers that this is like actually useful and novel? Like the concept took a lot of work to put into practice. It shows off something that has not been done before, but how practical is it, right? Because you have to kind of strike a balance between showing off something that's novel and something that's useful, right? Which is the eternal problem. And then I'm sure people that write grants and stuff are always struggling with this, right? You want to mess around, but you need to show that it's useful at the end of the day. So what I'm doing right now, and this is kind of off the slides, what I'm doing right now is experimenting and showing in what ways this score function is more powerful than the rest. Like in what applications does it actually really beat things out of the water, right? And there are some, which are pretty interesting. I can get into them at the very end. And here are the data on the yeast. Now, here's a little question. Can someone tell me that's something a little funky about the blue line that might be a little uncharacteristic? Or yeah, so like what's interesting is that its initial Q value is high, which basically means, say, okay, we care about things at 1% FDR, but if we cared about things at like 0.5% FDR, oh, we'd do terrible. We don't have any peptides to talk about, right? So why is that? And this is something that's pretty interesting and something I'll actually get people's opinions about because maybe someone actually knows something about this. And this is us on the left, Casanova, right? And these are all also decoys. So every single one of these is wrong. We should not be giving these high scores, yet we do. And these are the first couple decoys, high-scoring decoys for Tide, and these are the first couple high-scoring decoys for Casanova. We, for some reason, love the short peptides, and we score them really high even if they're wrong. Meanwhile, Tide's over here messing up all over the place, more evenly distributed, right? So there's something probably more fundamental going on with their mishaps, right? Which is why the curves are more natural. And then we have our thing where it's like, we screw up less overall, but we make some pretty critical mistakes early on. If we were to remove these mistakes somehow, it would really boost our performance, right?
What scoring do you use for, like, when you get the probabilities of a sequence? You get like the raw next token probability.
Yeah.
How do you convert that into a score?
Exactly. So this is exactly where I'm going. Currently, it's literally just averaging them, which is what actually seemed to work the best for the de novo sequencer, which is why was the initial score function for this. But turns out that has a bias towards shorter peptides. Now here's, I guess, my open question. First off, does it kind of make sense, like what the problem is? Like do you notice something unique about this list versus this list, right? So I guess my open problem, and this is where I kind of make a conversation, if you're given like basically a list of like numbers, like I don't know, something like that, I don't know, whatever the hell, how would you turn this into like one value in such a way that it doesn't result in shorter peptides inherently getting higher scores? So that's one of the open problems. Yeah.
I don't know if this works, but if you have any information from the aspect that somehow lets you know that this protein is not short.
Interesting.
Just get rid of all the proteins.
Yeah. So that filtering is usually like already done, and so the thing is like there's probably another short one that's actually correct, but this one probably beat it out by just like a little bit, right? Because it's short. And if it was longer, Clearly, the longer ones don't have that problem, and the reason is we have more information about the longer ones. We have like 20 or so probabilities, and if they're all right, that gives us a whole lot of confidence that that peptide is actually the correct one, right? Versus a short one, we only have 6 kind of checks, and then we're just kind of saying, okay, sounds good to me. So this is currently what I'm like working on. Like
Other methods don't have this problem
because I I I can't necessarily say why they don't have the problem, but I can definitely explain why ours has this problem more. I'm sure that like longer peptides have some kind of like difference to them. Like I mentioned, calibration is an issue.
you can do a you can just do a this is a case of it
Mhm.
might work,
Mhm.
such as ML. You could just do like a lambda value for what am I trying to say? Like what is it called? Ridge regression.
Interesting.
Like penalize shorter.
That's actually good you mentioned that. Not ridge regression, but there is a post processing tool for this kind of stuff made by the lab that employs a support vector machine, takes in some orthogonal information, and rescores the peptides, and it tends to solve problems like this, which means that actually we benefit a lot from that tool. Like if you apply that tool to us, the jump in accepted peptides is greater than the jump in accepted peptides for other score engines, right? But we want to kind of like show that we're good out of the box as well. So like like another thing we did is like take the logarithm of all the scores and then sum them, which ultimately becomes a multiplicative score. And since log is a what's that called? It's like that. It doesn't actually change positives and negatives. There's a name for that. I forget what it is.
Oh, wait, say it again.
It's like if I apply log to something, it preserves inequality. What's that thing called? It's on the tip of my tongue.
Eric, you studied math. A bit.
It's like the it's like a characteristic. It's like an attribute of a function that makes it such that like if you were to apply it to a set of values, they wouldn't like flip axes or anything. It's like preserve yeah preserve the order. Yeah.
I don't even know what you're talking about.
Like if I
If you have something that you can take the log of a function monotonic.
monotonically increasing. Yes, that's the one. So since log is monotonically increasing, you can take the sum of logs, and it will essentially act the same as like a multiplicative score. So that's one thing I'm trying. And then I'm also doing some like really weird like length normalization stuff.
I think you should just take the length of each one and let's see. We want these scores to be higher. Yeah, so just take the length of each one, multiply it by lambda, and figure out what lambda should be added to the score.
There's like a lot of cool creative ways to do this, and this is where like the research is open and it's like fun to experiment, but then you have deadlines and stuff, and it's like yeah. But I guess what I'm just trying to say is like this is a presentation on research. It's like open. Like I'm gonna wake up and like work on it tomorrow, and it's fun. It's in progress. There's a due date coming up for like a conference that I need to hit. I need results. I need to get off my ass and work, but it's kind of hard, you know. So let me go over like the last couple things, and then I'll wrap this up, and we can have a cool discussion about it. So one promising thing is that the overlap in peptides between like the other score functions and the database stuff and ours is very high, which is good because people have been using these tools for years and they've obviously resulted in plenty of discoveries. So we want to show that we'll get the same discoveries and more, which tends to be the case, right? You never want to say, yeah, we discover a completely different set of peptides. One of us is wrong, and they're gonna assume we're wrong because we're like the new dog on the block or something like that, right? So this plot essentially shows that what you want is all the red to be third quadrant, and then we want all the blues to be generally up in this quadrant, and then this leakage is somewhat okay. It'll take a second to like interpret maybe, but this is good. Trust me on that. And if you look at the raw overlap percentage, it's like 99%, which is what you want to see. Conclusion. These are the future experiments, and this is exactly what I was talking about. Yeah, I'm working with a PhD student, Justin. He's great. He's helping me a lot with this stuff. He's gonna take over for one of the search engines that I didn't want to mess with, so he's a G for that. And then we have like a bunch of other experiments lined up, and we actually had a meeting today, like literally what time? It was like 11 today, that like laid out the roadmap for the rest of the paper. So that's pretty cool. So this is like, if all things go well and I actually work this weekend, there'll be like some form of like a finished paper submitted to a conference, ISCB, which is pretty cool on this topic. So yeah, that's the presentation on the research I've been doing. I hope I'll be able to like actually give like another one later, like when things are complete, which would be super fun. But yeah, anyone have any questions on that? Yeah. Awesome. Maybe we can stop the recording now.